Welcome back everyone. In this video, we're going to continue our previous analysis. In the last session, we performed differential expression analysis and obtained our list of significant genes. That analysis tells us which genes show changes in expression, their log-fold change and statistical significance in different conditions like cancer and normal cells. But DG analysis doesn't tell us what these genes actually do at cellular or molecular levels. It only highlights that their expression levels differ between conditions. So, to understand the biological role of these genes, in other words, to identify which cellular functions or pathways are affected, we move to the next step of functional enrichment analysis. Functional enrichment helps us understand whether certain biological processes, molecular function, or cellular pathways are overrepresented in our list of DG compared to what would we expect by random chance. So in other words, instead of looking at single genes, we are now looking at functional patterns of genes, which pathways or processes are collectively affected in our experiment due to these gene expression changes. So there are two main approaches that we are covering: overrepresentation analysis and gene set enrichment analysis. The idea of overrepresentation analysis is pretty straightforward. It starts with a predefined list of significant genes like the DAGs we obtained. So we only work with the significant genes when we do overrepresentation analysis. So we then test if any functional categories such as biological processes or pathways are statistically overrepresented in that list of significant genes. ORA typically use statistical tests like Fisher extract test or hypogeometric test to measure significance. It is simple, fast, and work well when you have a clear threshold for significant genes. Gene set enrichment analysis, on the other hand, does not rely on any arbitrary threshold. Instead of taking only significant genes, it considers all genes ranked by their expression changes. For example, we can use different threshold like log-fold change or T statistic. So it then tests whether these predefined gene sets are tend to appear at the top or bottom of that ranked list. This approach is more sensitive, especially when changes are subtle or when you expect coordinated shift across pathways rather than sharp changes in individual genes. So to understand the difference, let's take a simple example. Think of it like a quick survey. Imagine we ask 100 students about their favorite drink and 20 says they love coffee. Now, when we look closer, we find that the most of coffee lovers are from a biology department. That tells us something meaningful, that the biology group is overrepresented among coffee lovers. So that's similar to overrepresentation analysis or ORA, where we only work with significant genes. On the other hand, if we didn't just pick a cutoff, but instead ranked all the students by how much they like coffee from most to least, and then check if biology students mostly appear at the top of that ranking list or not. That's more like gene set enrichment analysis. So to summarize, ORA works on a subset of significant genes, while GSE works on the entire ranked list of all genes. So enrichment analysis relies on the predefined gene set. A gene set is a collection of genes that share a common function, pathway, or regulation. For example, gene involved in a glycolysis from one gene set or genes involved in apoptosis from another. So these gene sets are curated from different databases. The two most commonly used databases are Gene Ontology and KEGG. They are almost standard in every transcriptomics or omic analysis. Gene Ontology provides a structured vocabulary describing a gene function across three main domains that are biological processes, like what the genes does in terms of biological objectives like cell division or immune responses. Next, we have molecular function, what the gene product does at the molecular level, such as ATP binding or enzyme activity, etc. And the third category is cellular component that address where the gene product is located, for example mitochondria or nucleus. So these geo terms are hierarchically structured. Broader parent terms encompasses multiple specific child terms. For example, here in this picture, suppose biosynthetic process is a specific child term under a broader categories related to carbohydrate or metabolic processes. So as we move up to the hierarchy, terms become progressively more general, eventually reaching a top level geo categories. These are the three main geo terms. And the other major resource is the KEGG. KEGG provides pathway, maps integrated genes, protein, and metabolites into biological networks. So it helps you understand how genes interact in broader biological systems like glycolysis, apoptosis, or cytokine signaling, etc. These pathways cover a wide range of biochemical processes that can be categorized into 7 broad categories. So simply put, GEO focuses on the function of individual genes. KEGG focuses on the interaction and pathways among these genes. We also have MSIG database, that is a molecular signature database. It is the one of the most widely used resource for gene set enrichment analysis. MSEG database provides collection of curated gene set derived from a different publication, experimental data, or computational analysis. Among these, the hallmark gene set collection is widely used. These hallmark sets summarize well-defined biological states like TNF alpha signaling, and also we have a list of C1 to C8 set of genes. It depends on what you need. So we can use them both for ORA and GSEA depending on the analysis approach. Now let's move to the coding part and start implementing enrichment analysis step by step. First, we will load the required libraries, cluster profiler org.hs and MSIG-GBR along with a few supporting packages. We use cluster profiler package which support both gene ontology and KEGG enrichment analysis. So to perform overrepresentation analysis, we only need a list of genes from our DEG results. For the demonstrating purpose, I will use the combined list of all significant genes, meaning both upregulated and downregulated. You can of course perform enrichment separately for upregulated and downregulated genes if you want to interpret direction specific pathways. It depends on how you want to debrief your data. So at this time for ORA, I will use both downregulated and upregulated genes. For running enrichment, first we need to convert our gene sample into entries ID. That's because some databases, particularly KEGG, don't recognize gene samples directly. Geo support both gene sample and entries ID, but KEGG specifically required either entries ID or ensemble identifiers. So it's a good general practice to perform this ID conversion at the start of your workflow. You have already seen this similar step earlier when we use map ID function to convert prob IDs to gene sample during annotation stage. This time we will use a similar approach but with a different mapping from gene samples to entries ID. First, let's extract all the gene samples from our DEG results, and here we will use the org.hs database. That's the human organism specific annotation package. If your dataset is from another species, you will need corresponding organism packages. For example, if your dataset is from mouse, you can use org.uppercase m and lowercase m database. Similarly, for Drosophila, you have a database uppercase D and lowercase m. You can also find a full list of available annotation packages is under Bioconductor annotation packages. At this time, we have keys as samples that are our gene samples, and the columns will be entries ID because we want to map gene sample to entries ID, and key types equals to samples. So if I run these lines, so at this time, entries ID column has been added to our DEG results. Next, we drop the missing entries ID from our data DEG results. So here you can see we don't have any missing IDs, missing entries ID at this time. Let's subset our gene group based on the regulation so that we have updated results. Results are same from our previous analysis, but at this time we have one additional column of entries ID. So for further analysis, we use this DEG up and down list of entries ID that contain all the upregulated and downregulated genes, but we remove non-significant genes from this list. So before moving forward for enrichment analysis, especially for overrepresentation analysis, we need to define a gene set. We need to define two important gene set that form the foundation of our statistical comparison. The first one is the background list, which represent all the genes that were tested in our differential analysis. This is essentially the full universe of genes that could have been detected in our experiment. So this list contains all the genes that are significant, upregulated, downregulated, or non-significant. Second one is the foreground list, which contain only the significant genes, the ones that are either upregulated or downregulated. So we use these two gene lists to determine whether certain biological pathways are overrepresented among the significant genes compared to the entire background gene. So here how we define them in R. So in this code, background gene represents the entire list of entire set of entries ID that were the part of our DEG results. And the significant gene contain only the genes that pass our significant threshold, that was adjusted p-value less than 0.05 and log fold change greater than 1. And now gene lists are ready for overrepresentation analysis. We perform geo-enrichment analysis by using this enrich geo function from cluster profiler. This function enrich geo uses a hypergeometric test to check whether the certain geoterms are enriched in our list of significant genes compared to all tested genes that are our background genes. So in this function, genes equals to significant genes. So the list should be into a character because entries IDs are in numeric form. So first we need to convert them into a character. Otherwise, this enrich go function will not work. And universe equals to background genes. In this case, all of our genes. And org.db is our database. We have key type entries ID. We are working with entries ID. So key type equals to entries ID. Ont equals to all. Ont means ontology. As I mentioned earlier, Geo has three main categories: biological processes, molecular functions, and cellular components. And we will be able to use these categories in this enrich function by specifying our ontology. So for biological processes, we can mention ont equals to BP, biological process. Similar for the molecular function as MF and cellular component as CC. This ont equals to all mean that we are using all of these three components in our enrichment analysis. So let's run it. It will take some time. It's done. Let's look at the geo enrichment results. Let's convert it into a data frame. Here you will see different columns: geo ID, gene description, ontology, geo ID and description, a gene ratio, that the number of gene enriched in a specific term divided by the number of input genes. Then we have background ratio, the number of available genes, a number of all genes in a specific term divided by the number of universal genes. So a gene ratio is the number of enriched gene in a specific term from our list and the number of input of genes. So this is the ratio of our significant genes divided by the our background genes that we provided. And in background ratio, this 390 is the number of specific all genes available in that geo term from gene ontology database, that is divided by the all the available universal gene of that database. So this is the ratio of the database. Then we have some other statistical analysis: z-score, p-adjusted value, q value, and gene IDs. Here you can see we provide the entries ID, but geoterm automatically provide us a gene IDs. Now we can visualize results using library enrich plot. We can plot a bar plot or dot plot. So here we can do a dot plot to visualize the result. It displays results on x-axis based on gene ratio, number of total enriched genes divided by the total number of inputs. So here the size of dot indicate the number of gene associated with that particular term. And the color gradient represent the statistical significance, that is p-adjusted value. The y-axis is the list of the geo term. The darker the color, the stronger the enrichment. Similarly, we can plot bar plot So the bar plot represent result in the form of count, number of genes. So the bar length indicates the number of genes contributing to each term. Similarly, we can split this enrichment result based on the ontology. So in this bar plot, we can specify our ontology split function. So we want to split our result based on the ontology column, that is this biological process, molecular function and cellular function. So if we run this line of command, now we have separate results for each category: biological process, cellular component and molecular function. You have lots of other visualization options with this enriched plot. You can check out for more detail regarding visualization. You can check out the cluster profiler P8. I will share the link for your reference in the description below. Next, we perform enrichment analysis for KEGG pathways, another fundamental database that widely used in nearly every omics or bioinformatics study. KEGG arrange genes into metabolic, signaling, and disease-related pathways, enabling us to connect our DEG results to specific biological mechanisms. Here we use enrich KEGG function. Here again genes equals to our significant genes, and the important thing we need to specify here we need to specify the organism for pathways, that is HSA for Homo sapien, for human, and key types are KEGG, that will be entries ID. So if we run these KEGG analysis is a start. So a KEGG enrichment analysis has been done. First, let's convert it into a data frame to check the results. KEGG as dot data frame. So here we get 9 enriched pathways. First category is metabolism, and this is the subcategory xenobiotic degradation and metabolism. Second enriched pathway is human disease that related to cancer. It means our analysis is correct as we are working with the cancer data set. So we have in our data set, we have enriched pathway cancer. Similarly, we have metabolism. Mostly we get enriched pathways related to a metabolism. We can also visualize the pathways that enlist the total number of our enriched pathway. This way we can perform overrepresentation analysis using gene ontology or KEGG pathway. So now after performing ORA with the GEO and KEGG, we can extend our analysis using Gene Set Enrichment Analysis. So unlike overrepresentation analysis, which require a predefined list of significant genes, gene set enrichment analysis uses all ranked genes by expression changing that allow us to the detection of pathways still but coordinated effects. Like we use log4 change to arrange our gene list. Cluster profiler provide various functions for gene set enrichment analysis. For example, you can perform gene set enrichment analysis with geoterm, with GSEA geo function to query gene ontology database. Similarly, we have function GSC KEGG to query KEGG database. Also, we have a universal function with this within this cluster profiler package, that is GSEA, Gene Set Enrichment Analysis. This is universal function. That means here we can use customize list of gene set or databases. So this function provide more flexibility that allow us to use custom pathway collections, gene sets. So this is specifically useful when we using curated sets from different databases. Like in this analysis, we will use Hallmark gene set from MSigDB database. Similarly, we have some other databases like Reactome. If you want to perform a specific collection for gene ontology, you can use GSC geo function. If you want to query KEGG, you can use this GSC KEGG function. And for customization, we will use this Gene Set Enrichment Analysis function. So the first thing we need to prepare our gene list. First, we rank our gene by using log-fold change. So in this way, the upregulated genes rank to top of the list and the downregulated will be on the bottom of our this gene rank list. And next, we need to retrieve hallmark gene set from MSig database. So for this, we use this MSig dbr function from the package. Here we need to specify two things: a species, that is Homo sapien, and category, that is H for hallmark. On MSig database, we have two categories, two species, Homo sapien and mouse. So we are working with human data and we have different categories, like we have a list of curated pathways. So here you can mention the categories accordingly, like C1, C2 for curated pathway, C5 gene ontology collection, or anything, any specific collection you need. So let's retrieve the hallmark gene set from the database. You can manually download the list from the database as well. So here we need a data frame with two columns only, entries ID and gene set name. Now we can perform gene set enrichment analysis using this GSEA function. Here our gene list is equals to gene rank, the entries ID of our data set, and the turn to gene equals to hallmark, that is our hallmark gene set from MSig database. So if I run this line of command, so this is how gene set enrichment analysis results look like. We have different columns, ID columns. The raw names are same as ID columns. Next, we have description of this pathway. Again, set size. Here we have this NES column is important. That is normalized enrichment score, that tells you the direction. So NES greater than 0, positive values are upregulated pathways. And the NES less than 0 or negative values means that these pathways are downregulated. That means these pathways are enriched in downregulated genes and positive ones are enriched in upregulated genes. Next, we have some other statistics, and now here the score enrichment is the entries ID. So we have results in the form of entries ID. These are the list of entries ID that enriched in that particular pathways. Okay, so the enrichment analysis is complete. We can visualize how gene contributing to each pathway and are distributed across the ranked gene list. First, let's start with the gene set GSEA plot. GSE plot function from enrich plot, which illustrate the running enrichment score for a specific pathway. So here in this function, we need to specify our results, gene set enrichment results. Then we are interested in looking gene set ID. So let's say this first one, gene set ID equals to hallmark bile acid metabolism. And if we run this line, so here this plot display how genes are associated with the selected pathways. So the x-axis represent all the genes ranked by their statistics, for example, a log-fold change, and the gene curve shows the running enrichment. So the interpretation is straightforward. If the vertical bars representing pathway cluster toward left side more, it means it indicates the upregulation of pathway genes. Conversely, if the bars are concentrated towards the right, lower part of the ranking list, pathway genes are downregulated. Let me show you with another example. First, let's plot one upregulated and downregulated pathways based on their NES results. So Gene Set Enrichment plot. GSC result. Now gene ID equals to, let's sort our result. First select positive value, that is 3.03 for the pathway, that is epithelial mesenchymal transition. So we will plot this pathway and plot it by their running score. Title of this plot will be epithelial mesenchymal transition. It shows that our analysis is correct. We are working with cancer data, so this pathway is upregulated. It makes some sense to our results. Now let's plot one downregulated pathway based on NES result. Select this negative one, that is hallmark of bile acid metabolism. Now let's combine them by using cowplot function. Cowplot, plot grid, plot 1, then plot 2 we want in column 1. So if I run this line of command, so now you can see the difference here between upregulated and downregulated pathways. The first plot shows the epithelial mesenchymal transition pathway, which most genes are upregulated here in the top of the list, while the second plot is bile acid metabolism that has a negative NES, indicated the downregulation, as most of the gene enriched at the bottom of the list below ranking of 15,000. Peak of the curve shows the maximum enrichment scores, highlighting where the pathway is overrepresented in the ranked gene list. So in this way you can visualize your result and present it. Another versatile plotting function is GSC plot 2, which allows more customization and support multiple gene set on the same figure. Like we have a function GSC plot 2. Here we need to specify our gene set enrichment analysis result. Then in gene ID column, we can specify the number of genes, number of pathways that we want to plot, like 1, 2, 3, or any number of pathway you want. And if I run this, here you can see now we have results of 3 pathways in a one plot. You can check out the distribution of their gene enrichment analysis. So these 3 all pathways are upregulated. Similarly, you can add the table showing p-adjusted value directly on the plot. Display p-value table along with pathway's name. So here we need to specify p-value table equals to true and run this line. So now along the name of each pathway we have their p-value and p-adjusted value. So in this way you can visualize your gene set enrichment analysis of results. Now we have completed our enrichment analysis. We perform overrepresentation analysis and gene set enrichment analysis. So let's summarize and organize all the results in a structured way for interpretation and reporting. First, let's convert the enrichment result object into a data frames so that we can easily manipulate and filter them later when we need it. Then now filter the results one by one. First, filter the geo result based on the adjusted p-value. We filter out our result based on their significance. So here p-adjusted value is less than 0.05. As we perform gene ontology on all of 3 geo terms, so let's separate out all of these geo terms. First, extract the top biological processes. We will extract top 10 biological processes enriched in our result based on their p-value. Next, extract the top molecular function, top 10 function, and top cellular component from our results, and combine all these top geo result in one data frame. So now we have top results based on their categories. Let's reorder this category column. Similarly, we can extract the significant KEGG pathway based on their p-value and significant gene set enrichment analysis result. And for gene set enrichment analysis, we can also subset our result based on NES values for upregulated and downregulated pathways separately. So in total, we have 37 pathways, but at this time we have result, we don't have gene samples, we have entries ID. So first let's convert them into a gene samples for more clarity of our results. Now here you can see we are mapping entries ID to a gene samples. So at this time we return result as 1:1 mapping between keys and column. So at this time our results are more cleaner. That's why we got 1:1 mapping. Rerun these code to incorporate changes in these subsets as well. So core enrichment enters ID with their respective gene samples as well for all result, for all significant GSC result. Similarly, we have gene sample for upregulated GSCs and for downregulated. So again, we have a long list of significant genes. So to summarize, or you can extract top pathways. Like if you want to extract top 10 pathway, you can do that as well. Now we have top 10 pathways from our result, and in last, let's check the summary of our overall result, what we have done so far. So let's create a data frame and calculate the total number of our results. So the overall summary shows the total number of significant terms for each analysis method, including geo. For geo, we have total significant 349 significant terms. For KEGG, we have total 9 pathways, significant pathway we get. For ORA, we don't have subcategories of upregulated and downregulated. We only got significant and non-significant results. But for GSE, we have significant results that are 37 total significant pathways. Among these 37, we have 26 pathways upregulated and 11 are downregulated. That's it for today's session. We have now completed our genomic analysis module.